So now we're going to have Sajja, Vinay, Kumar from Strand Life Sciences and Sam Pearlman, who will be talking about a lot of our modular infrastructure that we've been developing here for launching bioinformatics workflows. After that, we're going to go ahead and do the panel on AI. And then our last talk will be Jing Liu on the DeepCAH machine learning caller. And then we'll have time for some more networking. Yeah, Sajja, can you go ahead and start talking?
So hello everyone, this is Vinay. So today I'll be presenting on ConfigDB, MRD Analytical Pipeline Version Management at Myriad. So can we go to the next slide, Shane? So first I'll start with the current infrastructure. Then I'll be walking through the current challenges that we have been facing. From there on, I'll introduce the proposed solution, ConfigDB, a configuration service, and explain its role in improving the pipeline version management. I'll then also briefly touch on some technical aspects of the config service. After that, I'll outline the transition roadmap from S3 to ConfigDB. I'll then cover the analytical pipeline repository structure, followed by how we leverage GitHub workflow automation. And then finally, I'll wrap up with some key takeaways. Next slide, please. Let's first of all start with the current infrastructure. So currently, MRD assay retains module and versioning for both tumor profiling and ctDNA detection pipelines within a single GitHub repository. So within this repository, MRD analytical workflows, we maintain two key files. A version file which tracks the overall version of the analytical pipeline, and a configuration file modules.yml to track individual versions and other metadata of each module. So whenever any new version of a module is released, this configuration file is updated to reflect the new module version, and the version file is incremented to represent a new overall pipeline version. So once these changes are made on pull request creation, the PR merge to the updated config file is updated to the published to the staging S3 bucket, and on PR merge to mainline branch, it goes to production S3. So in both the cases, in the overall pipeline, the organized in S3 by the overall pipeline version. Next slide, please. So now let's look at several key challenges when managing both analytical pipelines together. So one major issue is versioning conflicts. Since both pipelines share the same repository, any pipeline that uses a version number ends up consuming the GitHub tag and Jira fix version. So this prevents the same tag or fix version from being reused for the other pipeline. So this also creates difficulties in the release process, as determining which tickets should be included in a specific release based on the GitHub version difference becomes unclear. Another complication is maintenance. We need a secondary main branch for one of the pipelines, which adds complexity and overhead to maintain the secondary branch whenever there are any infrastructure updates made to the primary main branch. So finally, there are also regulatory risks. Overlapping versions across pipelines within a single repository can lead to confusion in regulatory documentation, and they can also disrupt our existing established versioning practices. So these challenges highlight the need for independent pipeline version management. Next slide, please. So to address the challenges of managing both the pipelines in a single repository, so we propose decentralizing the repositories by splitting each pipeline into its own repository, allowing for independent versioning. On top of that, we also introduce ConfigDB, a centralized configuration management for our analytical pipelines that can be easily scalable to support additional pipelines in the future. So a ConfigDB simplifies infrastructure by eliminating the need for a separate S3 data stores for staging and production, while they also still support the separation of staging and production pipelines by using parameters. Uh, a ConfigDB also enhances auditability, tracking version promotions, records who promoted a version, and captures the exact release timestamp of the production version. I'll be talking about the config updates in the further slides. Next slide, please, Shane. So now let us walk through the comparison of the current infrastructure challenges and how a proposed solution of decentralization and ConfigDB will address them. So, by separating pipelines into individual repositories, we can assign version numbers, GitHub tags, and fix versions separately. So, this removes version conflicts, simplifies PR and release management, and it also reduces maintain overhead as we eliminate the need for secondary main branch in this case. And finally, we also mitigate regulatory risks with improved auditing capabilities. Next slide, please. Let's now briefly go over the technical aspects of the ConfigDB. So currently, ConfigDB is built using AWS Cloud Formation, composed of several AWS stacks, namely RDS, API, and Bastion. So the API stack is a serverless API exposed through the API gateway. So where each endpoint is backed by its own Lambda function, and the RDS stack provisions the Postgres database built upon SQL Alchemy models that stores all the version related information of all the pipelines. And the Bastion stack is a temporary stack used to interact with the database and to run database migrations, ensuring the schema stays up to date across deployments. So next slide, please. So let's now walk through the implementation roadmap for transitioning information from S3 to ConfigDB, starting with the foundation phase. So in this phase, we first create two new repositories, one for the tumor profiling and the other for the ctDNA detection, which allows us to have independent versioning. We then backfill all existing historical pipeline versions into their respective repositories by creating the configuration files that capture the relevant details for each pipeline version. So, moving to the automation phase, here we create GitHub workflows to automate how configurations are populated and how versions within the config DB are promoted directly from the repositories. So by making use of the created config files within the repositories, we backfill the config DB, ensuring we have the complete historical record. So next slide, please. So we then move on to the integration phase, where we update all the consumers of the config DB, like the MSM, to fetch appropriate pipeline version and configuration information directly from the config DB instead of the S3 buckets. So to support this, we use config DB client, which acts as a thin wrapper around the config service API, making it easy for all the consumers to interact with the config database. So finally, in the compliance phase, we update and finalize all the regulatory documentation to reflect the new analytical pipeline repositories. So next slide, please. So let's now take a look at how the new repositories are structured. So each analytical pipeline repository will contain a version file tracking the version of the current active analytical pipeline, and a configs folder that contains all configuration files, where each config file represents a specific pipeline version. So this file name itself indicates the pipeline version, and within each file, there is a mapping of all modules used in that pipeline and their respective versions. So the contents of these configuration files will be structured in the same way as the modules.yml file in the current infrastructure that exists in the current infrastructure. So next slide, please. So now let's take a look at the GitHub workflow automation used for publishing to ConfigDB. So whenever a new version config file is created and a pull request is opened in any of the newly created repositories, so the config file is first published to ConfigDB as a testing pipeline version. So from there on, the testing pipeline can be manually promoted to a validation pipeline at the discretion of the user. And once the PR is merged into the main branch, a GitHub tag is created and the version is promoted to an RUO release. And finally, any authorized users can also promote the version in ConfigDB to a production release version. Next slide, please. So, in summary, here are the key takeaways that we get. So, first, scalability. By introducing decentralization along with the ConfigDB, we can now support any number of analytical pipelines, and it also improves automation with GitHub workflows managing the entire version lifecycle end to end. And we also gain a full auditability with complete tracking of every version promotion and their release. And from a regulatory perspective, separating pipelines into individual repositories prevents version conflicts and removes regulatory ambiguity. And finally, it also simplifies maintenance as infrastructure changes are easier to manage without the need for a secondary main branch. Next slide, please. Okay, yeah, thank you. That will be all from my side. I think I'll now pass over to Sam from here.
Everyone hear me? Whoa. Hi, everyone. I'm Sam Pearlman. I'm a bioinformatics software engineer for Myriad Genetics and also on the scientific solutions team. I'm going to talk today about the developer experience and accelerating the pace of development. So you just got to hear Vinay describe our new ConfigDB service, which manages our pipelines. And here is a large image of our data management system. I will not be describing this tonight, but my excellent colleagues, Eric Anderson and Benjamin Weishviel, gave a full talk on this at the September 10th BioInformatics. So you can go back and I think post-register to watch that. So these are two big pillars of our development environment. But I think there's a third one that's been missing, and that is the developer experience. And that is one of my teammates, Mittens, working very hard on bioinformatics. So I will be running through what the developer experience was when we started these projects. I'll describe the FRAC build management tool, plus our reorganization app. And I'll finish up with our SPAR system for running analyses and generating reports. So DNA Nexus is our cloud compute partner, and this platform has been extremely valuable to us in our development lifecycle. They are HIPAA compliant, and we operate in a heavily regulated space. It's scalable. We run tens of thousands of analyses per year and generate petabytes of data. But one area we found we could innovate is in speeding up our development cycle and automating as much of the programming and code generation to execution time processes. This is because scientists want to focus on their innovations. They do not want to spend their time dealing with software infrastructure. Before we did this work, the build cycle included many manual steps. So to illustrate, I have a short 12-minute video of what it takes to build one of our pipelines. I'm just kidding. Nobody wants to see that. But I think that illustrates how little our developers want to spend 12 minutes, making one small code change and pushing it through to building a workflow. To illustrate it, first let's say the developer updates their code. They then have to build a Docker image. They then have to upload that image to DNA Nexus. And where they upload it is a source of common errors. Once they upload it, they get a new file ID for that Docker image. Go back to their code, put that image reference in their workflow files, then use DX compiler to build the workflow, and finally manually run that workflow either by the command line interface or through the web interface that DNA Nexus provides. This is not ideal. So we came up with FRAC, the Fast Rebuild Automation Kit. And that is definitely what FRAC stands for. It is not a Battlestar Galactica reference. So with this new system in place, you update your code, but instead of manually specifying where your Docker images live, it's templatized using Jinja. So you would specify Docker images and your environment, and then you basically just hit go. The command is actually frac build everything, and you use YAML files to configure projects you want things to go to. And then frac takes over and takes your code and builds the workflow. It smart builds your Docker images. It detects whether or not any code has changed in a particular environment and only builds the environments that need building. It then uploads the Docker images to pre-configured DNA Nexus projects and gets their new file IDs. It then goes back and renders the WDL, the workflow language, with the correct file IDs. And you can see on the left, the templatized WDL has been replaced with the actual location of the image. Then DX compiles the rendered WDL into a workflow. And where that workflow lives is also stored in a pre-configured project. As an extra bonus, the FRAC system builds into the workflows a very last step called reorg or reorganization. That takes all your output files and arranges them into logical groups any way you like. But what it also does is send that output file and data locations to our DMS, our data management system, which I briefly showed you a picture of earlier, one of our big pillars of software development. So you kind of get that for free, and the developer does not need to do anything once the initial config file is created. The next system in our environment, which has made everything much easier for our computational scientists, is SPAR, Sequencing Pipeline Analysis Regression. So after the build process, you need to actually execute your workflows and get results. So we built SPAR as a way of automating running the analyses, comparing the results of different code changes in regression style tests. And all you need to do is choose your code branch or commit, choose one or more gold standard sequencing runs from the box, and again, just hit go. And it will build your workflow if you have not previously built it from that commit. It will run the analysis with pre-configured inputs and then compare the results it gets after completion to the stored reference results. It will then present any differences for review in a report. And if you're making code changes that result in changes to outputs, you can then update the gold standard reference analysis for future runs to be compared to. It also gives improved visibility and traceability. As I said, we are heavily regulated and we often need to know exactly what commit a particular run came from. This is a screenshot of one of the reports. So in this particular case, all the calls were identical, but there were differences in the metrics, and you can configure exactly what gets compared. But if there were call differences, you can dive in and look at exactly what calls were different. Here, the genotypes were different for 2 different calls in the VCFs. So to summarize, I went through what our initial developer experience was like, and it was not very enjoyable. We then built the FRAC management tool plus the reorg app that automatically hooks into our data management system, and then the SPAR analysis and reporting system to actually run the workflows that are built by FRAC. And these systems have helped enable us to form a scientific solutions team, which its goal is to rapidly develop and deploy new assay pipelines throughout our research and development organization. For acknowledgements, I wanted to thank my entire team, including Dana Case, our Director of Bioinformatics, Shane Brubaker, our Engineering Manager, and of course, our computational scientist partners. They very much help us drive our innovations in this area. And of course, Mittens. And that is it. I guess we're not going to do questions right now. Not going to do questions right now. Thank you, everyone.